In this video, we will study about what are dicot plant and what is the internal structure of primary stem of dicot plant. As we know, in angiosperms, there are two kinds of plants, dicot and monocot. Dicot mean dicotyledonous and monocot mean monocotyledonous. Now, what is cotyledonous? Cotyledonous is presence of here we can see this is a dicot seed and we can see here are two cotyledons. But in case of monocot, there will be only one cotyledon. So if two cotyledon, it is called as dicot plant. If one cotyledon, this is called as monocot plant. Examples of dicot plants are so many, but I'll mention few of them like mango, neem, all the pulses, gram seeds, Kabuli chana, black chana, kidney beans, rajma. This is also sunflower, because here we will be discussing anatomy of sunflower stem. So as we discussed, presence of two cotyledon is one characteristics of dicot plant. Now this is the gram seed. Now this is dicot plant I have shown with the help of diagram. We can see here. This is the dicot plant, and this part of the plant is root system and this part is shoot system. Shoot system is, this is the main stem and these are the branches and these are the leaves borne on branches, and this is the axillary bud. So another characteristic feature of dicot plant is by which we can identify just externally by seeing the plant, a presence of reticulate venation in case of leaves. Like here in this diagram I have tried to show how network is being produced, right? Now this is network of the veins present in the leaves. So this is the network. I have not completed the diagram, but here also same network is there. So this is called as reticulate venation, mean network of veins. Second characteristic feature is presence of taproot system, right? This is not a fibrous root system, this is taproot system. Taproot system is, this is the main root, these are secondary roots, these are tertiary roots, and these are root hairs. So this taproot system, presence of reticulate venation and presence of two cotyledons. These are main characteristics of the dicot plant. Now if we take its stem, for this we are selecting sunflower plant. Now this is the stem of the sunflower. These are leaves borne on sunflower stem. This is the flower, but we are talking about stem only. So we'll just remove the leaves and we'll take the stem. And when we take the stem and we can cut its section, this section is called as transverse section, right? This can be cut with the help of razor, blade or microtome. So when we cut the section, first thing when we see under the microscope, we will see that all the vascular bundles, they are arranged in the form of a ring. Like this is stem and these are the vascular bundles which are arranged in the form of a ring. There is distinction between this part is epidermis, this part is cortex, then this is the vascular zone and this is the pith zone. Now this characteristic feature, that is vascular bundles, they are arranged in the form of a ring. This is a very important feature to study the internal structure of the dicot stem. Now if we take one part of the stem like this and we see its cellular details, I mean cell formation details, because if we see this diagram, this is diagrammatic. I mean this is not cellular, this is diagrammatic. But if we take one part of it and study its cellular detail, and we will see that now this is the sunflower stem, right, in detail. We can see here this is the outermost layer called as epidermis. Now this is made up of just parenchymatous cells like this, and this is protected by cuticle. Cuticle is made up of cutin. This will protect the internal organ. At certain places this may be having openings called as stoma, and these are hair-like structures borne on the epidermis. They are multicellular in nature, mean made up of many cells. Inner to epidermis, this part is cortex. Now cortex is of two types. This is the outer cortex and this part is inner cortex. Outer cortex is made up of collenchymatous cortex. Now what is collenchymatous cortex? This is one cell, this is another cell, this is another cell. You can see here. At at certain places, at angles, thickenings are there. So because of these thickenings, this is called as collenchymatous cortex. To know more about collenchymatous cortex, you can watch my video of tissues. The link will be shared in the description box. Now this part is made up of parenchymatous cortex. Parenchymatous cortex simply cells, they are thin walled. They are enclosing intercellular spaces. Now this is the intercellular space for gaseous exchange. At certain places this may have oil duct. Sunflower oil, we have heard of it. So this is being produced in the form of oil canals, and these oil canals are filled with oil, and this is called as oil cavity. Now this part is called as endodermis. Now this is separating cortex from the vascular zone, right? Now this is the vascular zone and this is the endodermis. Sometime this endodermis may be filled with starch, so this is called as starch sheath. Now inner to endodermis is pericycle. Now this zone, which is shown with the red color, and this is quite thick wall. Now this is present above the vascular bundle. So this is called as sclerenchymatous pericycle. And this zone, which is falling between the vascular bundle, this is called as parenchymatous pericycle. So inner to endodermis is pericycle. This is sclerenchymatous. This is parenchymatous. So this is also called as heterogeneous in nature. Now these are the vascular strands, which are arranged in the form of ring, like we have earlier seen in the previous slide. And this thin zone of the parenchyma present between the vascular bundle will be called as medullary rays. And this part, which is again made up of parenchyma zones, parenchyma cells, they are quite thin wall. Again, to study more about parenchyma cells, you can watch my video of tissues. Now this part is called as pith, which is present in the center. So this is about cellular detail of the transverse section of stem of sunflower. And if we see the vascular bundle in detail, we can see here. Now this is the endodermis, which is separating the vascular zone. Now, each vascular bundle is conjoint. Conjoint means this has phloem as well as xylem. When both are present, this word is used. This is called as conjoint. And collateral, when they are present at the same radius in the vascular bundle, this is called as collateral. And this is a strip of cambium which is present between the xylem and phloem. This is again made up of parenchymatous tissue, and this is meristematic in function. Meristematic means when it can divide and redivide, this will cut off secondary phloem toward outer side and secondary xylem toward the inner side. Secondary is, this is the primary phloem, this is primary xylem. Later on, when growth take place isodiametrically, secondary phloem and secondary xylem are cut, so that those secondary growth will be occurring with the help of this cambium. And because of the presence of cambium in between phloem and xylem, this is called as open type. Now we can see here xylem element, this bigger element which are bigger in size, these are metaxylem, and which are smaller in size, they will be called as protoxylem, because this part is facing toward the outer side and this part is facing toward the inner side of the stem. This condition is called as end-arc condition. And in between, this will be the xylem parenchyma, and this is about xylem, cambium and phloem. Now we will study about detailed outline of the structure of dicot stem, like epidermis. We know this is the outermost layer. This is made up of parenchymatous tissue, and this protects the internal organs, as well as this also prevents excessive loss of water, that is excessive transpiration, because this is the outermost layer, so this can perceive the light. So this is the site of light perception. Now next is about cortex. As we discussed earlier, this is made up of outer cortex and inner cortex. Outer cortex is collenchymatous, inner cortex is parenchymatous. Like we discussed, this part, when it is thickened at angles, this is called as collenchymatous cortex. And when this is thin walled, enclosing intercellular spaces, this is called as parenchymatous cortex. The function of cortex is, this provides, if it is like collenchymatous, and this will provide certain strength to the stem, mechanical strength to the stem, and parenchymatous tissue will provide flexibility. I mean, this can move easily. This is called as flexibility. And if this zone has chlorophyll, then this will help in photosynthesis. And all the intercellular spaces present in the cortex, they help in gaseous exchange, and very important function also, that extra food material will be stored in the cortex in the form of starch grains and lipids. Now, next layer we know that this is endodermis. This is delimiting the or separating the cortical zone from the vascular tissue. This may be rich in starch grain. This is called as starch sheath. Now, next is pericycle. As we discussed earlier, pericycle is sclerenchymatous. This will provide mechanical strength, and parenchymatous, this will function as food storage tissue, because two types of sclerenchymatous as well as parenchymatous pericycle is there. This is called as heterogeneous pericycle. Now, vascular strand, we know that vascular bundles, they are arranged in the form of a ring, and their shape is wedge shape, and they are arranged like, if you see the stem like this, and these are vascular bundles, and this part is called as pith. So, they are present around the pith. So, this arrangement is also called as U-stele type, right. So, this arrangement is called as U-stele because they are present around the pith. As we discussed, both xylem and phloem form joint when present at the same radius, collateral, and when they have a cambium in between, this is called as open type. And because of the presence of cambium, this can show the secondary growth. And when cambium is not there, vascular bundle, they are called as closed, and no secondary growth will take place like in monocot stem. So, this is all about anatomy and cellular detail of the internal structure of the dicot stem of sunflower. Now, if we see phloem is made up of sieve tubes, companion cells, phloem parenchyma and phloem fibers. Like these are 4 types of cells which make the phloem. Then xylem. Xylem is made up of tracheids, vessels, xylem parenchyma and xylem fibers. And we know, we have discussed just now, protoxylem element which are smaller in size, and bigger element, they are metaxylem element. So, they are broader and they are smaller. Now, strip of cambium we have already discussed. This is a thin area present between the vascular bundle. This is vascular bundle, and this is the thin strip present between the vascular bundle. So, this is also called as intravesicular cambium, which helps in the secondary growth. Now, medullary rays, we know that this is the stem and these are the vascular bundle, and in between this parenchymatous zone, this is called as medullary rays, and this is important for lateral translocation of food and minerals to the plant. So, this is present between the vascular bundle called as medullary rays, helping in radial translocation of food and minerals. Now, in the center of the stem, like this is stem and these are the vascular bundle, and this part is called as pith, and this is made up of parenchyma cells which may be oval, they may be polygonal like this, or they may be rounded like this. So, this will help in storage of food in the form of starch grain or sometimes in the form of lipid bodies. So, this is all about anatomy of dicot stem. Thank you for watching my video. If you have any questions, you can ask me in the comment box. Thank you for watching.